everyone. My name is Sonu Paswan, roll number 62, and today I am going to cover an analytical device named Voltammetry. This is a device which is used in various fields including analytical chemistry, material science, and electrochemistry to characterize and analyze properties including varying its electrochemical reversibility, determining the number of electron transfer during its oxidation or reduction, and determining the equilibrium constant in the coupled reaction. I will start the brief description of these topics. Later on, I'll cover whole details in one by one. So, voltammetry is an electrochemical technique where the current at electrode is measured as a function of applied voltage. So, basically, it is a device which used to measure the current across the electrode under applied voltage. This technique is particularly useful for analyzing electroactive species which can be cation or anion. So this device is used to produce the currents under applied voltage, and with the help of current we are able to know the concentration of analytes by the following relation. That is, current is proportional to concentration of analytes. And this technique is used for both qualitative and quantitative analysis of analytes. So whenever there is a analyte present in our solution, then we always get a measured current, and with the help of this measured current we are able to know the intensity of this concentration of these analytes. Now here the experimental setup and component during our experiment. So instrumental setup. This is a device which contains three types of electrode. One is working electrode, reference electrode, and auxiliary electrodes, and ammeter which is measuring the currents, and a voltmeter which is measuring the voltage across the electrode, and a variable resistance which is used to divide the potential difference across the electrodes, and a battery which will supply the voltage across the electrode. I will cover each and every electrode in detailed form in upcoming slides. A voltmeter, potential difference between voltmeter is used to measure potential difference between working reference electrodes, and ammeter measures the current between working and counter electrodes, and a battery provides a necessary voltage to running this system. Here the different setup in the laboratory. Now I am covering some basic terminology which is used in voltammetry during the experimental setup. The voltammogram. Voltammogram is the graph obtained from voltammetric experiment showing the relationship between voltage and current. This is a result we get from these experiments at the last. This is a graph between voltage and currents. With the help of this, we will analyze the analytes concentration, and both we use both quantitative and qualitative analysis with the help of these graphs. And basically there are two types of current generated during voltammetry experiments. One is Faradic current and another is non-Faradic currents. So Faradic current is produced due to the oxidation or reduction of an analyte is known as Faradic currents. There are two types of Faradic currents. One is cathodic current and another is anodic current. So cathodic current is a current generated due to the reduction of analytes, and the anodic current is the current generated due to the oxidation of analytes. So this is the current generated due to the Faradic current, and non-Faradic current is the current produced not due to the oxidation or reduction. This is often arising from the other process at the electrode surface. Now I am going to explain the internal mechanism, what will happen inside the electrochemical cell, how the mass will be transferred from one place to another place. So basically there are three modes of mass transfer in electrochemical cells. One is diffusion, migration, and convection. So diffusion so diffusion is a diffusion is a transfer of mass due to the concentration difference. Here the concentration difference is the driving force of this type of mass transfer. So at the electrodes, we see that there is a reduction happening. That's why the near the electrodes, the analytes reduces their electrons. So this will reduce the concentration at electrode. That's why the concentration, the mass will be transferred from bulk solution to electrodes. In this way, the diffusion mass transfer will take place in the electrochemical cells. So when electric current is passed in analytes, the analytes near the electrode either oxidize or reduce. As ions at the electrode surface react, their concentration decrease, causing ions to move from higher concentration area to lower concentration area. Now come to the migration. The movement of cation ion toward the positively charged electrode when voltage is applied. So this is a mass transfer happen by the movement of ions toward and opposite to the electrodes. The anions move toward the anodes and while cation move toward the cathodes. The extent of ion movement depends on the applied voltage. So intensity of the movement of these ions depend on the voltage capacities. Now come to the convection. Mass transfer will take place at convection mode. The movement of analyzed ions in response to mechanical force such as stirring the solution. In controlled experiment, convection and migration is minimized to focus on the diffusion currents. So we are only concerned about the diffusion current. We are not concerned about the convection or migration current. So that's why we only know how the diffusion by mass transfer take place and the current generated due to the diffusion mass transfer. The expression of calculating the diffusion currents are there, in which diffusion current is I is equal to NFAD, C bulk minus C surface by del. Here N is the number of electron transferred in the reaction, F is a Faraday constant, and A electrode area, and D is the diffusion coefficient of species, and C is the bulk concentration of solution, and Cx is the concentration of analytes at the electrode surface, and del is the thickness of diffusion layers. So in this way, we are calculating the diffusion current across the electrodes under the applied voltage. Now I will come to the instrumentation of this voltammetry device. The instrumentation of this voltammetry. Basically, this voltammetry device consists of three type of components. One is electrodes, potentiostates, and voltammetric cell solution. So I am covering these three instrumentation in these slides. So I will start with the electrodes. And there are three type of electrodes. One of them are working electrodes. The working electrode is where the actual oxidation or reduction of the analytes occurs, and the potential difference is continuously changed between working electrode and reference electrode. A voltmeter is placed between electrode and reference electrode to measure the such change in voltage. Here many examples of working electrodes are: one is hanging drop mercury electrode, suitable for very long concentration analysis. This is a type of working electrode which used to analyze a low concentration range of uh, analysis at the ppb to ppm level. Another is static drop mic electrodes. In this we are used to measure the concentration of analyte at very low concentration range. Another is rotating disc electrodes. And the material used for the construction of this type of electrode are gold, graphites, and glassy carbons. And another electrode is reference electrode. Reference electrode is the electrode which is used to measure the potential difference of the working electrodes. And here the potential difference of this electrode is fixed. And the main function of this electrode is to maintain a stable and non-potential, allowing for a precise control of working electrode's potential. By providing a fixed reference potential, it enables the potential state to measure the exact potential difference of the working electrode. And the example of this electrode are polymer electrodes, mercury-based reference electrode, silver, silver and silver chloride electrodes. And the last one is auxiliary electrode. The main function of this auxiliary electrode is to complete the circuit by allowing the flow of current between the working electrode and the auxiliary electrode. It does not measure the potential but serve to balance the reaction at working electrode. Current flow, how the current will be flow, and in which electrodes, in between electrodes, the current will be starting to flow from which electrode to which electrode. I will the current flow between the working electrode and auxiliary electrode allowing the accurate measurement of analyzed response. And the material used for this type of electrode construction is typically made of inert material like platinum, classic carbon due to their high conductivity and resistance to corrosion. Now the last one is voltammetric cell solution. The voltammetric cell solution basically made up of supporting electrolytes and analytes. Supporting electrolytes, a sub that stabilizes the solution environment and assists in measurements. And the analytes, the substance being analyzed for its electrochemical behavior. Analyte is the substance that we want to measure the concentration of this substance in voltammetric experiments. Now some characteristic of supporting electrolytes. The supporting electrolytes main purpose is reduce the migration, lower the effect of ions migration, minimize the interface with the analyte's movements, and decrease the resistance, lower the solution resistance, allowing for the better current flow, accurate measurement. And there are some basic characteristics of these electrolytes, and these should be chemically inert, high ionic conductivity, and increased selectivity, pH adjustment. This should be chemically inert because when we applied this supporting electrolytes in our analytes, then they need to be interacted with the solution of our actual. We want to measure the concentration of these analytes. And high ionic conductivity provide a better current between these two electrodes whenever you want to current flow from electrode to electrode. And high selectivity will provide a good efficiency in current measurement. And the pH adjustment provide a good for our equipment safety. It will reduce the corrosion. Now I come to the result of this experiment, that is voltammogram. So it is a graph plotted between voltage, that is on the x-axis, and the current on the y-axis. Typically show the S curve as we saw in the diagram. So the current generated at the initial state, that is residual current. Here is a from here to here, this is a residual current. This is a current generated without analytes due to some impurity present in the solutions. And another is diffusion currents. Diffusion current range is from here to here, that is continuously increasing. This is generated by oxidation or reduction of analytes of ions. Higher the concentration of analytes, more will be the diffusion currents. So higher the concentration, more will be the current generated in the our cell. And this is a limiting current, that is The maximum current, limiting current is the maximum current that can be achieved and dependent on the concentration of analyte. These are all the current is basically depend on the concentration of analytes. So for formula of calculating this limiting current is I is equal to K into Ca, where I is the limiting current, K is the constant, and Ca is the concentration of analytes. So here we clearly see that the concentration of analyte is directly proportional to the current generated due to these analytes. Now there are many applications in industrial as well as pharmaceuticals. Some applications are these are these analyses are used to monitoring the environmental issues like detection of pollutant. Voltammetry can detect the trace amount of pollutants such as heavy metal, pesticide, and organic compound in water and soils. And also used in pharmaceutical and clinical analysis like drug testing. Voltammetry techniques are employed in the pharmaceutical industry for analysis of drug and active pharmaceutical ingredient, often detecting trace impurity and measuring oxidation reduction property of molecules. And also used to measure the blood glucose monitoring. Voltammetry is the principle behind some blood glucose meter which measure the glucose level in blood through enzymatic reaction that produce a current proportional to glucose concentration. And in biomedical sensor, it is also used in biomedical sensor used in detection of biomedical molecules such as DNA, RNA, protein, specific ion in body fluids. Voltammetric sensor can help diagnose and monitoring various diseases. And this is the technique is also used in food and beverage industry. Like to control the quality, voltammetry can help to detect additives, preservatives, and contaminants, including pesticides and heavy metals, ensuring the safety of quality of food product. And also used to monitoring the fermentation process. In beverage production, voltammetry is used to monitor fermentation by tracking chain in certain compounds. So these are the application and working of this voltammetric device. Thank you. This is all about from myself. Thank you for watching.